Thank you. If you could just get seated, we can get started. It's great to see everybody back for an excellent session this afternoon on conservation and restoration genetics. And to kick off, we've got Jason Bragg from the Botanic Garden and Domain Trust in Sydney, giving us an overview of genetics to inform restoration and conservation of eucalypts.
Can everyone hear me okay? No? Yes. Okay, so I'm going to be talking mostly about landscape genomics today. However, I'll also talk a little bit about some of our related activities in Myrtaceae genomics and a study of introgression that have made use of the Eucalyptus grandis reference genome that we're here to celebrate. Also, though, I wanted to flag that Susan Rutherford will be giving a talk about some of the other work going on at the Botanic Gardens and Domain Trust in conservation genomics of eucalypts. So, as I said, this talk's mainly about landscape genomics, and so just to give a quick definition of that, a landscape genomic study is one where, say, we sample a species across its range on this idealised map here, and we sample some number of populations. We can measure the amount of differentiation or genetic differentness between those populations and study patterns in variation and use those patterns to try to make inferences about processes. So frequently what we see is if we look at the levels of differentiation between pairs of populations and consider them as, say, a function of spatial distance, we see some sort of monotonic increase, some relatively simple relationship, and we might refer to this as isolation by distance. This line might be steeper, for instance, if there's less migration across the landscape per generation. However, this isn't what we always see. Sometimes we see strong discontinuities in patterns of genetic variation. And so what I mean by that is, whereas in isolation by distance we see the highest levels of differentiation between things that are far apart, with a big discontinuity, say at a river or something like that, we might also see high levels of differentiation over a relatively short spatial scale. And these might be due to a barrier to gene flow that perhaps is extant, or a period of evolution in isolation and two lineages coming back into secondary contact, or something like that. And so, so we're doing multi-species landscape genomics, and there are lots of basic questions that we're interested in, such as how different species have responded to a common environmental template. But the overarching motivation for what I'm going to tell you about today is doing this research to inform collections for restoration. So say on this landscape, idealised landscape we were just looking at before, if someone wanted to perform revegetation at this site, where should they get the seeds from? We've also heard this morning, it's been a theme that frequently collections have emphasised local provenances. There's an increasing shift towards trying to go broader with collections. In each case, what we want to do is try to figure out how broadly one can go without running into sharp discontinuities or exceeding what we think might be a reasonable threshold level of differentiation to mix. And so this can vary quite strikingly among species or across space within species. So if we were collecting for a site here, we could potentially go quite a long way south and not run into a big differentiation. But as soon as we hit say the river in the north here, we might acquire much more differentiated seed. So we're undertaking a study to measure this in dozens of species in a project called Restore and Renew, which is being led by Maurizio Rossetto at the Royal Botanic Gardens in Sydney, and is being undertaken by a large team of researchers, including Marlene, Hannah, Peter and Susan Rutherford, who's here and presenting this afternoon. So as I mentioned, we're doing multi-species landscape genomics for many species. For each species, we're collecting at between 20 and 35 sites across New South Wales. At each site, we're collecting 6 unrelated individuals, and in each individual, genotyping thousands of SNPs with DartSeq. So now I want to sort of step through how we how we make inferences from these data and how we present that information to the public via the Restore and Renew website, which is now live online with a modest number, 8 species on there currently, but a number more ready to go and others in various different stages of the analysis pipeline and still collection and sequencing. So these are data for species Calicoma serratifolia. I won't go into the details currently, other than that these are data collected by Samantha Yap and Maurizio Rossetto. These are the sampling locations, and this is a plot of pairwise differentiation between populations as a function of spatial distance. And what we want to do is turn this into something where someone can click on an arbitrary location on a map, having selected this species, and be given a prediction for how far they can go without exceeding a threshold level of differentiation from the location that they've chosen. So where we think it's safe to go and collect seeds. So the way we're doing that is fitting a generalized dissimilarity model. And so there we use a statistical model of pairwise differentiation, here measured as FST, as a function of, in this case, spatial distance. But we're also, in some cases, introducing other explanatory variables to the model. I just want to also make the note that GDM, or generalized dissimilarity modelling, was developed by Ferrier and others and has been applied previously to these kinds of problems, including by a paper by Megan Supple et al. In Eucalyptus meliodora. A couple of quick notes on this. So it's perfectly, we should expect that in some cases we will see strong discontinuities in genetic variation on the landscape at locations that don't really bear any sort of relation to a measurable property of the landscape, that lineages have separated, say under historical climatic conditions, and just come back together where they come back together. And so in some cases we can't actually necessarily use an environmental variable as a predictor, and so there we've been estimating coefficients of membership to ancestral populations, say through structure, or we're using SNMF, and putting those vectors of membership to ancestral populations into the GDMs. So just to work through this example for Calicoma serratifolia a little bit further. So in this case, we estimated a GDM and got a reasonably good statistical fit just using spatial distance as the single predictor variable. So then we say, for instance, we take the blue diamond there as the location where we choose to do restoration. We can then predict under that GDM what the level of differentiation is to all other grid cells within this sort of bounded area, which represents a buffered hull around the sampling locations. And we see that, as we'd expect, because only distances in this model, the predicted levels of FST are smallest around the diamond and grow larger as we move away. Then if we apply a threshold and mask out all of the cells that exceed that threshold, in this case we're using 0.3, though we're still quite actively thinking about what the best threshold might be for all cases. We get a prediction for the area within which we think that you would collect seeds that are less than some threshold, genetically different than from your restoration site. So we've this information is being served on the Restore and Renew website. So you can go to the website, choose a location. You'll then get a drop-down list of the species that have sort of those buffered hulls that that site falls within. So they're the species that you can choose. It'll then run a prediction under the GDM that's been prepared for that species and give you this area. This unmasked area here is the area within which we think you'll be able to collect material that does not exceed some level of differentiation from your site. I should say here that I think this is, rather within keeping with the idea that that we should be sort of encouraging some collections to promote diversity, so not purely local but also extra local. So while this would seem to be saying keep it within an area, it's sort of I guess what we'd also encourage is collecting from multiple trees and potentially broadly or just at multiple locations within the suggested area, and we make some sort of suggestions about that kind of thing in some sort of materials that are generated with the prediction. But I'll now just step through another example that I think sort of serves to illustrate how this is kind of useful. If we look at what we saw for one species and then sort of work through another species for the same location. So the story in Acacia suaveolens is rather more complex in that we see levels of differentiation that are much higher overall, but we also see some reasonably strong population stratification. So 3 structure groups seems to work pretty well, and the Sydney Basin sort of seems to be quite a different lineage, but even more broadly, it seems like we start encountering different lineages as soon as we move away from the coast, the coastal margin in any substantial way. And so here, the best fitting model, the best fitting GDM, had spatial distance and difference in elevation as the 2 predictor variables. And so then if we step through and choose the same site in the Restore and Renew web tool for Acacia suaveolens as we did for Calicoma, and we have the same level of zoom, the area where we think you can collect and not exceed some threshold level of differentiation is much, much smaller. Also, because it's not just spatial distance in there, which sort of gives you just a constant radius, the shapes also become much more irregular because they're also responding to elevation and not just spatial distance. So this seems like a good place to mention that eucalypts are also coming to the Restore and Renew website, and the work on eucalypts is being led here by Susan Rutherford, and so there's, I think, like 15 species are being collected currently. The first couple that have sort of come through complete collections and data analysis are a couple that are mainly found in the Sydney region. They're Angophora hispida and Corymbia eximia. In both cases, we see relatively low levels of differentiation. In fact, Angophora hispida looks pretty much panmictic. And Corymbia eximia also we see very low levels of differentiation that really don't ever exceed the kinds of thresholds that we'd be likely to apply. So in those cases, the recommendation would probably be collect your seed quite widely within the range. So now I'll just briefly mention a couple of other projects that have been using the Eucalyptus genome, as I mentioned. So in one case, we sort of are using this as an opportunity to think a bit more broadly about how we might cope with hybridization and introgression in the context of the Restore and Renew study. So Eucalyptus saligna is a great Restore and Renew species. Its range is it's distributed between sort of south coast of New South Wales just up into Queensland. But we also know that in parts of its range, it's sympatric with species with which it's known to hybridize. These include Eucalyptus denii and Eucalyptus grandis. Also Eucalyptus botryoides, which I've left out of this just for now because we've mostly been sampling north of Sydney, and it seems to hybridize extensively south of Sydney with botryoides. So if this is the distribution of saligna, we see that there are areas where it occurs in sympatry with Eucalyptus denii, which has this really disjunct distribution, which in fact at different stages have been recognised as different species. It's also sympatric with Eucalyptus grandis quite often in the south of the range of grandis. I thought kind of one of the interesting things about that was that Eucalyptus saligna occupies quite a lot of the climate envelope of Eucalyptus denii and Eucalyptus grandis. And so I think in the longer term there might be some interesting questions there about whether or not there's adaptive introgression. Is saligna borrowing genes from the other two species to cope with life in those actually pretty different environments? But in the first instance, we're mostly interested in trying to better understand between these eucalypts how much introgression is going on, how recently did it occur. And so we've been sampling saligna by itself in the absence of grandis and denii, and in sympatry with each of denii and grandis. And we're trying to do so with a study design that will allow things like 4 population tests of introgression and ABABABA analyses when we do some whole genome resequencing of a selected number of samples. But for now, and those, for instance, ABABABA tests were explained brilliantly by Rose yesterday. But for now, I'll just show you some preliminary analyses. And I guess the thing that I found quite surprising here is, consistent with what's been observed previously, we see some F1 hybrids in the field. But despite the hybridization that we do see, we see 3 pretty discrete things that I guess a priori I was probably expecting them to be much less well differentiated than they in fact appear to be, apart from a few sort of reasonably heavily admixed individuals and a couple of F1 hybrids that we sampled. So the last thing I wanted to mention, just really briefly, is that in case it's at all useful, we've generated a couple of draft genome assemblies for a couple of other Myrtaceae species, which, and of course the Eucalyptus genome is helping a lot there as a reference for sort of understanding what's going on in those. And so our motivation here was that myrtle rust is impacting a number of species of Myrtaceae quite heavily. So we wanted to generate draft reference genomes for a couple of species that show relatively high susceptibility to myrtle rust, but also relatively high levels of variation in their susceptibility of myrtle rust, with the idea that these might be useful for studying the spatial variation in myrtle rust resistance, and also potentially for doing GWAS to try to identify markers associated with resistance. And we've proposed to do that with Rich Edwards and Justin Borovitz. So just a few details on these assemblies. We are therefore Rhodamnia argentea, which shows quite a lot of intraspecific variation in rust resistance, but is also quite closely related to Rhodamnia rubescens, which has become critically endangered due to myrtle rust, and for Syzygium oleosum. Okay, so finally I just want to thank the large number of people who've collaborated on the Restore and Renew project, the work on blue gums, and those Myrtaceae assemblies. Thank you.
Jason. One, do you have climate layers in the Restore and Renew, the isolation by environment? And second, how easily can other people put into Restore and Renew all the other samples that are coming?
Great question. I totally forgot to mention that. So we do not have climate in the GDMs at this stage, so we're thinking about sort of doing GDMs with climate, but then we'd sort of probably have another box down here that would be, do you want to see what this looks like and explain the consequences of that. What I totally forgot to mention though is that the blue area here is the area that is currently climate matched to the site that you've chosen. If you click in this box here, you'll see the area that is climate matched for predicted climate 50 years into the future. So great question. And on adding the additional species, please talk to us. Yeah, excited to talk about that.
So, how do you think you'll establish this threshold, and how different is it going to be with different species, and what are the factors that will affect it?
That's a great question. So I think in the I guess the question, the really tough thing is knowing, in fact, what is too differentiated, and that does seem like something. What is the risk of outbreeding depression, and I guess is the bottom line question. And so I guess what we really need to do is look at what evidence there is that outbreeding depression varies systematically among species according to their traits. Are there any traits that might be predictive of that? In the first instance, we set the threshold probably a little bit too high at FST equals 0.3. I guess, you could probably make a reasonably hand-wavy argument that an FST of 0.2 represents something about sort of 1 migration per generation, and above that you're sort of moving towards lineage divergence. But I think that's sort of a misapplication of the pairwise FSTs that we've calculated here. So I guess the bottom line is there's probably no way really to know except to do the experiments. And in the absence of that, we just need a good, useful, rule of thumb. And if anyone has any suggestions, we'd love to hear them. But we've designed it so that when a species goes in, the threshold is handed in as a parameter for that species. So we can adjust that in the future if just according to the best available information at the time.
Thank you. So our next speaker is Linda Baldwin from the Australian National Biodiversity Research Centre at CSIRO in the National Collections. And she's going to talk to us about Eucalyptus meliodora. Lots of information.
Lots of stuff about meliodora.
An important tree in the east.
Thanks, Margaret. So you've seen this species a lot this conference, which is great. Warms my heart. It's my favourite eucalypt. And a lot of my work's been dealing with helping practitioners understand what they're working with. So for restoration practitioners, there's lots of decisions in the field that need to be made. How many trees? Where from, where to, what's my site prep, what's my dollar value, and so trying to help them understand how they can do this a little bit more efficiently is a key outcome. So just going to walk you through the research I've been doing for about a decade now on this species. So just a little bit of background. So this is actually, there are lots of iconic animals, don't have many iconic plants, but actually this could be one of them. If you talk to farmers, they'll often say, do you want to see the photo of my granddad who parked his horse under our yellow box near the farmhouse? So they do resonate with this species. So it's quite useful when you're trying to ask them to do things with you. It's valuable for shade, for stock, for honey producers. It's a really important species. And it also provides sort of habitat connectivity, especially, I think Rose and I had exactly the same pictures of the regent honeyeater, so very rare species, and the superb parrot. Yes. So like most eucalypts, it's long-lived. We think it flowers around every 2 years, but that may have shifted in the last few years, but we don't have any hard evidence. We do know for some early work at Wagga that isolated trees produce less seed that germinates more poorly than woodland trees, and we think there are a vast range of pollinators, but no clear one is significant. It's also part of threatened box grassy woodland communities, which used to live here across Australia, very extensive, but it's now part of a critically endangered federally listed community, as well as endangered ecological communities in a state and a territory. Oh, wrong button. So it's got a broad distribution, but it's now highly depleted. And I just want to acknowledge Rowan Melick has done all this modelling, and for Carsten, later on I'll tell you the variables. So I put them in, seeing as that was a question this morning. I thought, I better get those straight into my talk. So I just want you to remember this as you're going through. This is what these woodlands used to look like. They were extensive up and down the east of Australia. And that's kind of what we're trying to think of when we're talking about the past of this species. So we know that 90% of the grassy box woodlands have been lost since European settlement. We know that half of the remaining 10%, so that's 5%, don't actually meet the minimum conditions for protection, so they're at high risk of further loss under the EPBC Act. And so there's obviously a really high priority for restoration of this species, and that's where I come in. And so Rowan, we'll get more to this later on, but he modelled distribution at the LGM, present distribution, and you can see that it has shifted. There's a lot more green on the map. And those small red dots are part of the big study we did that you saw a little bit of Megan's results earlier on today when Justin talked. So this was an interesting study. So I wanted to actually do a sweep of the whole species distribution but had no money. So invoked citizen scientists, and actually it was incredibly valuable to work with them because they tell you lots of stuff that you don't know and you can't see in the field for yourself. So we actually did an EOI process where we asked them to provide us with lots of information about their collecting experience. Had they ever collected Eucalyptus meliodora before? How good were their skills at identification? And then we also asked a few questions about how we're going to do it because we didn't want people falling off Land Rovers when they were collecting, so we need to have a few health and safety requirements undertaken. But basically we managed to put out an EOI. We had more than 50 people respond. We got more than 50 sites collected, but in this study we took it down to 32 because we could consistently get more than 20 trees, and overall the whole distribution we ended up with 850 trees in total. We also had some from WA because it went national, the EOI went national, and we had someone from near where I come from in WA put their hand up and go, oh, I've had them growing on my property for 15 years. So we sent out a collecting kit with instructions about how to collect, and we waited for the leaves to come back. We also collected seed. Some of that seed's been used, but some of it's still sitting there waiting for me to find a student to actually work with the seed. Megan's done a bit of work on it, which is great, but there's still more to be done. So we genotyped with 7 SSRs. I'm sorry, it's not thousands of SNPs, but please remember this is nearly 8 years ago now. But we did have sort of 8 to 26 alleles, so we were confident that the diversity at the time was reasonable. So just to give you an idea of allelic richness and expected heterozygosity, there are a few sites where allelic richness is quite low. I put crosses on them. That's probably not the ideal sort of symbol. That doesn't mean they're very bad. It just means that they're lower. But they are lower than some of the other sites. This is the WA site. It's got just as much allelic richness as many of the others. And then this is the Queensland site at the other end. And I've just colour-coded them between sites because it was just easier for me to visualise where these might be sitting in the landscape, were there any patterns associated with where they sat down that gradient from Queensland down into Victoria. And heterozygosity didn't really show us anything different. So from a practitioner's point of view, that's quite good. It doesn't look like there are low levels of genetic diversity that would preclude any of these sites being used as a foundation for a restoration project. In terms of genetic structure, so one of the things we were looking at was whether there are clear demarcations between different zones that might say don't move seed beyond this because it's got a different evolutionary history. I quickly threw in our FST value after I saw Margaret's talk this morning because I know it was within the range of what she was talking about, which is great. Our principal components, they account for a bit of the variation but not huge amounts. And if you look at this, it basically tells you that stuff's distributed everywhere. All the populations are kind of mixed up. And if you look at structure, K equals 4, so 4 genetic clusters, but with no real clear pattern of that in a spatial context across the east coast where this is distributed. So that wasn't really helpful, except if you think about this is we've actually sampled pre-fragmentation primarily. So this is kind of a representation of what that big box woodland was pre-European serious fragmentation in the landscape, because we actually deliberately sampled very old trees as part of the sampling. We were very keen to understand what was the structure pre-farming landscape change across the distribution. So that kind of makes sense. This is just the same structure plot at an individual level, and you can see it's a morass. I mean, this is, I think, the beast of eucalypts that we work with. They're all really complex and complicated, and got all these, as we know from Rose's work, hybrids being thrown at everywhere. You know, F1s, F2s, back crosses. We've got lots going on in some of these populations. And then this is just you saw this morning with Justin. So, you know, was that what we saw in the structure plots simply because our markers weren't differentiating the populations enough? And then with Megan's work, nearly 10,000 SNPs, no, that doesn't seem to be the case. This seems to be representative of this species because these are all the individuals, and there doesn't seem to be any clear separation or structure amongst that either. So that brings us into the present, because one of the things that's been happening is, you know, the last 20 to 30 years we've been doing a lot of restoration with this species, and I was very keen to understand, we know seed collection practices in the past were quite different, to the point where collectors would sometimes go to the same tree year after year because it was known to be a good producer, and so that became the basis for a lot of restoration projects in a lot of places. So, you know, somewhere around Canberra, for example, the same tree may have set up, you know, 10 or 20 different plots. So effectively it's one tree placed around the landscape 20 times. So with the help of Greening Australia, they were curious to understand this. Was this an issue for them? Given that they've invested a lot of taxpayers' money on behalf of taxpayers, they've done a lot of restoration. How good was that restoration, and what's its likely long-term persistence? So we chose, the good thing about Greening Australia is they have meticulous seed records, and so they chose sites around Canberra, so 5 sites around Canberra that had Meliodora in them, and we could go back and actually genotype those sites. They do have slightly different configurations for planting. That's restoration, that's the system that we work with. So they're different sizes, they're co-planted with different eucalypt species, they're sort of planted at different times. So effectively it's not a replicate experiment, but we decided to press on anyway, and we thought, well, the worst it could be is that they all react individually, and that's the story that we've got. The reality was they didn't react individually, which meant that I could actually collapse a lot of the data down. And so the idea for this study was to understand genetic diversity, and we kind of put a boundary on it. So we chose a restoration site, and we were all interested in understanding diversity in this, as well as the scattered trees around a site, as well as the seed crop that was being produced by the restored trees. We also just did a quick mating system study to confirm it was a mixed mater. And then the other thing I was very curious about was pollen movement. So it's one thing to put restoration back, it's another thing for it to be effective and persist. And so pollen dispersal is an important part of that long-term persistence. So we're interested here in understanding the rate of selfing in restored trees, the amount of pollen coming from scattered trees into the restored site, the amount of pollen that was being just transferred amongst the restored trees, and how much was coming from an exclusion zone of 250 metres. So we're kind of turning that longer distance dispersal. So able to collapse all the data down into 3 categories. So scattered trees, restored trees, and seed produced by restored trees, and these are the 2 allelic richness and expected heterozygosity. And what we see is that the scattered trees, of which there are far fewer, actually have higher levels of genetic diversity than the restored trees or the seed that the restored trees are producing. Now that's a pretty big concern given all the money that's been put into these restoration sites. If these aren't as good as this, what does that mean for population persistence? And the real challenge, I think, is that Phil Gibbons wrote a really nice paper. If you've not read Phil from ANU, Phil Gibbons wrote a very nice paper about scattered trees globally, and they're falling out rapidly. So you can go to somewhere like the Dehesa or the Oaklands or our agricultural lands, and scattered trees are declining rapidly. And they're expected to fall out somewhere in the next 150 to 180 years. And so that means that these restored sites, without these in the landscape, are actually in effect genetically poorer once the scattered trees leave the landscape. So that's a concern, and that actually now helps Greening Australia think about what they need to do to go back and reinforce these, including collecting seed from those scattered trees to put back in so that they're creating that next generation and boosting genetic diversity. The other part was to figure out where the pollen was dispersing. In this little graphic, the size of the arrows indicates where most of the pollen is coming from. And so very little selfing at most of the sites. A fair amount's coming from the scattered trees, which totally blew out my hypothesis. I thought the restored trees would be doing most of the pollinating because they're the dominant number of trees. That's not the case. The scattered trees are doing more pollinating than restored trees are. That could be because of their age. They may not yet be mature enough to actually compete with the pollen from the scattered trees, or it may be that they're all quite related and the preference is for outcross pollen from the scattered trees. What was really surprising though was that by far the most of the pollen was coming from beyond that exclusion zone. So in nearby woodland remnants, some of which we know are 1 kilometre away. And that also fits with Margaret's data about dispersal, and Eucalyptus wandoo, she knows, is going around 1 kilometre at least as well. So we do know that something is moving pollen quite large distances and quite a lot of it in these 5 landscapes. So the other challenge for Greening Australia was that getting enough seed. So it's one thing to know where you want your seed from. It's another thing to actually have enough seed to do something with it. And so we kind of did this study back to front. It's not quite how you might design a study, but this is what we did. So they had 20 seed lots and they wanted to build 2 seed production areas because they knew if they could build them in 10 to 15 years' time, they would have a good supply of seed. So we grew. So I got the seed, we grew it up, we genotyped it, and any seed that was left over we sent back to Greening Australia because it's very precious. We sent it back to them to grow on for their seed production areas. And so we planted it out. So there's 2 sites, there's 240 trees at each site. They're not trials, they're not built like provenance trials, they're particularly seed production areas. And we're tracking the growth. We've done 2 growth periods. We've done 3 growth periods. We've analysed 2. We're about to do another one soon. And basically, what we've found so far is there was no difference. There was a small difference for survival for seed lots. So these 3 that are arrowed actually didn't survive as well. We've got no difference in survival between the 2 seed production areas. No difference in height amongst the seed lots. There were height differences between the 2 seed production areas, and they're very different sites, which is kind of what we'd expect. In terms of genetic diversity, unlike the big study where we had 4 clusters, we actually had 8, which is really interesting. And when I first saw this, I was quite disturbed. I think when you go to the seed production areas, though, you realise that this is a real mix. So in those seed production areas, I think we've got misidentifications, we've got hybrids, and then we've got regular Meliodora. So I'm not surprised that this, again, is a real mix of what we've got. And then we also did some analysis with Rodney Dyer's pop graph, and these are the predictions from pop graph about where dispersal is occurring across the zone that we collected seed from. So that suggests that seed is genes are going quite large distances and possibly between species, potentially. And I just wanted to put this up. This is some more work that Megan did in her paper that shows you the diversity of what we're dealing with in this species. So these were just representative leaves from maternal lines that she'd stressed by drought. And this is yellow box. This is the morphological variation in yellow box. So it's not surprising that people find it hard to identify in the field. So where are we going with yellow box? So back to the modelling. So last glacial maximum today, and these are the parameters that Rowan did all the modelling with. So we then took it a step further to go, so what's happening 2050 and 2090? You know, most people are doing these things. And that doesn't paint a good picture for this species. But then the picture is much more complicated. So, you know, we all talk about, oh, species are going to track climate change and adapt and whatever. So I started thinking about this. Species can't track climate change in the southeast of the corner of this continent because land use prevents it. You cannot move these things to new environments. There is just nowhere for them to actually fit because there's like 20,000 plant species, you know. If you imagine, even if we're trying to move a couple of hundred into these spaces, if you've modelled where they might be able to live and put land use on it, it's just not going to happen. So I think that's where conferences like this, we're going to have to help these things live where they live now. I mean, that's going to be the real challenge, is because we're not going to be able to move everything into a new space. There just isn't a space for it to move to. And so I think that brings up lots of discussions for conservation and management into the future. Just a quick summary. So I think the provenance study sort of tells us that there are signals of the broad distribution that was there pre-European settlement. There's a lack of clear spatial structure. That's not unusual. It's been found in a couple of other species. And it looks like low genetic diversity is not going to hinder restoration practitioners in terms of choosing sites, as long as they look for those larger sites. I must say I've got a scattered tree that looks just the same in terms of genetic diversity as, in terms of its seed crop, as large populations. So there's lots of interesting things with dispersal. Restoration projects, potentially those done in the last 20 to 30 years, may have limited long-term persistence because of their limited genetic diversity. But remnant vegetation, scattered trees are probably going to be supplementing that in terms of pollen while they're still in the landscape. It means we need to look after scattered trees and remnant vegetation. And I think there is this still big undetermined morass of hybridisation, what's the species boundary, and clear identification issues for this species. And the future, I think there are real limited opportunities for this species in particular to track with climate change because there's no space for it to go to. So in situ conservation and management is going to be key to helping this stay in the landscape. Done.
Thank you, Linda. We're a little bit over time, so if people want to have questions for Linda, they can talk to her at the break. And next we've got Rebecca Jordan from CSIRO, recently down here in Tasmania, going to talk about Eucalyptus microcarpa, another pretty important tree in restoration for these in Australia.
Cheers. Thanks, Margaret. Thanks, everyone. So I came across a really lovely quote in a conservation genetics text that really resonated with me by Fred van Dyke. It was essentially saying that the aim of conservation genetics is to preserve species not as a static form, but as a dynamic entity capable of adapting and evolving to change over time. And so I'm really interested in how we can use genomics to inform conservation management and how we can think about capturing that genetic diversity to help create that dynamic capacity in our natural and restored systems to help with that long-term sustainability. And so some of the work I'm going to be talking about today is looking at, in particular, restoration, as this session suggests, and how we can use genomics to assess current restoration practices and think about what we might do into the future. So some of this stuff has been really nicely covered, so I'll just go over really briefly. But revegetation is a really important component of conservation management, especially in our highly modified landscapes. As has already been mentioned, in southeastern Australia, we've had a lot of clearing for agriculture, so we now have a lot of habitat loss, and what's there is highly fragmented. Often restoration is considered successful based on the presence of trees. However, if we're thinking about that long-term sustainability, it's not just the presence that's going to be important, it's going to be thinking about that dynamic capacity for those populations to be sustainable into the future. And as Dot pointed out really nicely this morning, and has been mentioned a couple of times, traditionally we've been thinking about capturing local diversity in our revegetation plantings, but thinking under changing conditions in particular, this is starting to shift. To think about collecting more widely across the landscape to capture genetic diversity generally into our revegetation practices, or using our knowledge of adaptation within eucalypts to be collecting along environmental gradients or matching to predicted future climates within the landscape. But using these is predicated on really understanding that genetic diversity across the landscape in the first place. And so this is where this small part of the project that I'm going to be talking about was wanting to know what is the pattern of genetic diversity across the landscape and how well have we actually captured that to date in our restoration plantings. And so looking at a number of levels, one at that really broad landscape level, those broad landscape patterns, at the site level, within sites, how well do we capture diversity? But then using the extra power that we've got with genomics, can we tease down into the genome level and see what variation we're capturing there? So I'm loving the box gum grassy woodland theme that we've just got going, especially today. So to add to the collection, this is Eucalyptus microcarpa, grey box, again across that southeastern corner of the mainland, agricultural area, extensively cleared, highly used in revegetation plantings to ameliorate that habitat loss. So made a really nice test case for some of these questions. So at 7 locations, I sampled a natural remnant as well as a revegetation site. And again, using DartSeek, as many others, had about 13,000 SNPs to compare diversity between these pairs of sites. So starting firstly at that really broad landscape level that Justin introduced, wherever he's disappeared really nicely earlier. This is a little bit of a busy picture, but I just want you to take away a couple of key things. One is looking at the colours. So this is looking at each of the individuals and how related they are across the landscape. The 7 different colours represent the 7 locations that I sampled, and what we see is that cline across the landscape. So we've got across PC1 is a south to north cline, and PC2 is an east-west cline. So we're seeing that pattern. This species doesn't really have any really distinct geographic barriers across its range, and so we're seeing that just slow change, that cline in genetic variation across the landscape. What we also see as well is the crosses within there are our individuals from revegetation sites. And we notice that generally they tend to cluster with their local geographic area, suggesting that local provenancing has been used for these revegetation plantings. I'll just put a little note in here at this point in time as well, there was next to no information about where the seed came from these revegetation plantings, so it's a little bit unknown. But it seems that broadly they've come from a local-ish geographic area. So that was looking at individuals. We then wanted to look at the sites themselves and think about the lovely graph that Jason had up earlier about how related those sites are compared to the geographic distance between them. Again, what we see, so the dark dots are our natural sites, and we see that lovely cline. So at those sites that are closer together are more similar, those sites that are further away are more dissimilar. And so suggesting that we do have that really nice cline of variation across the landscape. When we look at the crosses, though, at our revegetation sites, we see that this hasn't been captured. Our revegetation sites are more different from each other for any geographic distance than we would see between natural sites, and they're more different than to the natural sites as well. So that cline, that pattern of genetic variation across the landscape, which may also include patterns of adaptive variation, doesn't seem to be replicated in our revegetation sites. So if we then turn to looking into the site and how much diversity we find in a site, this is expected heterozygosity as a measure of diversity. We've got the 7 locations across the bottom, and again just focusing on the black dots being the natural sites and the crosses being the revegetation sites. What we see is that overall our revegetation sites have lower genetic diversity than our natural sites. However, this varies from location to location. You'll see in the Rock and Crowther over to the right, they have very similar levels of diversity in the revegetation site compared to the natural site. What we also see is where there is lower diversity in our revegetation, it's not a big decrease in diversity. It's only about 7% lower than our natural sites. So it's suggesting that we do have some capacity to actually possibly capture good variation in our revegetation sites. It then begs the question, though, if we have similar-ish levels of diversity within the site, and if our individuals are similar to the broad geographic area that we saw in that initial PCA right at the start, what's driving that differentiation that we saw between sites that means they're not capturing that overall pattern of landscape diversity? And this is where genomics really comes to the fore to be able to dive in more detail. So this is just a different representation of that previous graph I've shown you. It's comparing the level of site diversity in the revegetation site compared to the natural site. So the red bar is showing a decrease in diversity in the re-veg compared to the natural. And so you can see each of those has varying degrees of lower diversity, with, again, the rock and crowd having very similar levels of diversity. But that's a genome-wide average. Given the number of SNPs we had, we were able to break that down across the 11 chromosomes. When we look at the 11 individual chromosomes, what we find is that this change is really, really uneven. There are some chromosomes that have a greater loss of diversity, some that have lower, some that even have higher diversity in our revegetation sites compared to our natural sites. So this goes some way to possibly explaining that differentiation that we saw, that even though the overall level of diversity is the same, the actual variation across the genome seems to be different in our revegetation sites. So just sort of summarising this and thinking about where we go forward from this data suggests that the genetic diversity we capture in revegetation is approximately similar to natural sites. However, those landscape-wide patterns of diversity don't seem to be captured in our revegetation, and there are differences across the genome in that genetic diversity. So as we go forward, we've been seeing some really great talks today about understanding adaptation and areas of the genome that might be important for adaptation. And a complementary study to this that I did was looking at adaptive signatures in Eucalyptus microcarpa, and this is just a genome map with the coloured dots indicating adaptive signatures with, again, temperature, precipitation, aridity, similar variables to what we've seen. So as we get to understand those differences across the genome and possibly which regions may be more or less important now and into the future, we may be able to start to look at our revegetation in more detail. Can we look at adaptive versus neutral differences in genetic diversity between the two? And if there are differences, what does this mean for the future adaptability or that dynamic capacity of our restoration going into the future? And to tie these two together and to think back to that original slide showing the different ways that we could think about capturing seed across our landscape, I like to think about these and genetics more generally for restoration as being able to form where and how widely we should be capturing seed. So where, in terms of adaptation, where do we need to be capturing in the landscape to make sure we have good adaptive variation, but also that how widely, what Jason was talking about, where can we go across the landscape to make sure we just generally have good genetic variation to enhance that dynamic capacity in our revegetation plantings going forward. And so with that, I'd just like to acknowledge a whole heap of people who helped me with bioinformatics and similarly a lot of land management as well as Greening Australia who helped with access to various revegetation sites as well as other sites around and our funding bodies. So thank you very much.
We have got a couple of minutes for...
Excellent. Yeah, that's what I thought.
I think what you've shown there is that the revegetation, the seeds collected for revegetation go through extreme bottleneck, that you're actually showing this up in heterozygosity. It's an insensitive responder to bottlenecks. If you looked at loss of allele diversity, you've shown much bigger differences. Heterozygosity responds to much slower to small population sizes. So what you've got here really is a condemnation of the seed collection practices that we used for restoration plantings at that time. It's the same as in the previous talk. You're looking at an extreme bottleneck.
Yeah, absolutely. I did look at some other metrics when you're talking about number of alleles. This is SNP, so it's obviously biallelic data, so that they'll only have two. So,
but you would have got it from.
I think you're. I have to disagree with you. Like this is the most extreme total amount of genetic diversity we've ever seen, and going from 7 to 8% is just a minor dip. Like vegetation versus no vegetation. I guess what I'm saying is I'm worried that because it's not perfect, it's not exactly natural, that restoration practitioners are going to do it around for 4 years. Exactly where should I collect it? And I'm
Well, I couldn't disagree with you more.
Yeah, we're on exactly the opposite.
It's easy to get a few extra mother trees.
Sure it is,
If
but
people
it's
know
harder
what
to
the
like
process
do
is.
something. You know, we have to get trees
Yeah,
back
but
in the landscape.
If it's easy to do it properly.
I think it's an education.
Any other comments?
No, that's all right. I was just jumping into it. Yeah, no, that's all right. And it is a really interesting point.
We're never going to get it perfect. We just have to get trees in the ground.
I'll just jump in and just really quickly say what I was going to with being biallelic markers, so sort of number of alleles, this is kind of only 2, but what I did look at was the number of polymorphic loci within a site, so the ones that actually had multiple alleles, if you like, given that you could possibly have 2. And there was a slight decrease again, but again, it wasn't necessarily like this huge, massive decrease that we would sort of expect.
But you're sampling chromosomal segments, you're not sampling alleles here in this process. It's one generation.
Yes, yeah.
Right, thank you, Rebecca.
90% full.
Yeah.
Thanks, Rebecca. And our next speaker is Susan Rutherford from the Botanic Gardens in Sydney, and she's back here talking about population genomics and hybridisation and genetic swamping in a rare species.
So I'm going to be talking about managing the risk of genetic swamping in a rare and restricted tree using genomics. So I've been working on eucalypts for a little while, and I did my PhD on the green ash eucalypts. And as I was writing up my thesis, my supervisor from the gardens came to me and said, "Do you want to work on a project on hybridisation in Eucalyptus tetrapleura and other ironbarks?" And absolutely loving eucalypts, I of course said yes. So I ended up starting to work on that. And, hybridisation is a really important process in the evolution of many lineages. It generates genetic recombinations, it contributes to phenotypic plasticity, and we also think that it also enables some organisms to adapt to new environments. However, hybridisation can also have negative impacts on biodiversity. For example, in cases where a rare species is being placed at increased risk of genetic extinction by a more common species through genetic swamping. And we've seen a lot in the last few days about how recent advances in genomic methods are enabling us to address long-standing questions in evolution and biology. And the same is with hybridisation. So some of the questions in hybridisation can be addressed using these advanced techniques. So Eucalyptus tetrapleura is a really interesting species because it is distinctive in that it has 4 buds on the sides, 4 ribs, on the sides of its buds and its fruits. And it's quite restricted. It only occurs in a 40 by 100 kilometre area around Grafton on the north coast of New South Wales. And to put this into perspective, I have a distribution of co-occurring species, so species that co-occur with Eucalyptus tetrapleura, Eucalyptus fibrosa and Eucalyptus sidrifloa, which are far more widespread. And this is images of Eucalyptus fibrosa and Eucalyptus sidrifloa. And you can see they differ from Eucalyptus tetrapleura in that they have smooth buds and smooth fruits. Eucalyptus fibrosa is distinctive though because it's got these elongated caps on the buds. So Eucalyptus fusiformis, it doesn't co-occur with Eucalyptus tetrapleura, but based on morphology, it is thought to be the most closely related ironbark to Eucalyptus fusiformis. And Eucalyptus tetrapleura, and you can see that it does have similar buds and fruits, but they just don't have the ribs. And in a study by Woodhams, Dott, Beck-Jones, and Dean Nicole, they found using dart markers in a phylogeny that Eucalyptus tetrapleura and Eucalyptus fusiformis were sister species, so this supported the morphology. And also consistent with the morphological analysis, they found that fibrosa and sidrifloa were more distantly related to Eucalyptus tetrapleura. So Eucalyptus tetrapleura is listed as vulnerable under both New South Wales state and federal legislation. And it's threatened by a number of processes such as agriculture, timber harvesting, road construction, and grazing of seedlings and excessively frequent fires. And in more recent years, it's actually been cleared quite heavily in the central populations to upgrade the Pacific Highway. And this included an area at Wells Crossing where they actually cleared through where the type specimen of the species was found. And so after clearing the land, RMS came to the Botanic Gardens and said, oh, can you guys do a genetic study now on Eucalyptus tetrapleura to give us advice on how to regenerate the species? So we went in, and so the aim of our research was to examine the population genetic structure of Eucalyptus tetrapleura to aid conservation efforts. And so we tried to address the following questions. What are the patterns of gene flow and genetic differentiation within and among populations of Eucalyptus tetrapleura and closely related co-occurring ironbark species? Where are the purest populations of Eucalyptus tetrapleura? And is there evidence of genetic swamping of this species by more common ironbarks? And what management actions should be implemented now in revegetation efforts? So we collected across the known range of Eucalyptus tetrapleura, so there was our sampling sites. And we also collected from co-occurring species that were more widespread, like Fibrosa and Sidrifloa. And to provide cross-specific controls, we also collected Sidrifloa and Fibrosa from down near Sydney. We also collected Eucalyptus fusiformis. However, Eucalyptus fusiformis doesn't co-occur with tetrapleura, so we had to go to sites around Coffs Harbour, so at Bongal Bongal, to collect Eucalyptus fusiformis. And there's a photograph of Peter collecting an ironbark at Castlereagh Nature Reserve. So we collected our leaf samples, we sent them off to Diversity Race Technology, and where they were extracted for DNA and DART-seq analysis was performed. So we got our DART data set back, and I won't go into too much detail about DARTs because a lot of people have been talking about DARTs. So we got over 42,000 SNPs for 242 samples, and basically 5 of those samples had greater than 50% missing data. So we eliminated those from our analysis. And this is a split tree of the full DART data set. And we found that most of the samples of Eucalyptus fibrosa clustered together. Eucalyptus sidrifloa, most of those samples clustered together, and this actually included Eucalyptus fusiformis, which was a bit of a surprise for us. And most of the Eucalyptus tetrapleura samples clustered around here. And we also had samples that were intermediate between fibrosa and tetrapleura on the split tree, and samples between sidrifloa and tetrapleura on the split tree. So we then cut the data down based on purity. So we chose the markers that were the highest quality. And in order to do that, we deleted samples that had reproducibility less than 98%, as well as SNPs that had less than 10% missing. Did I say SNPs for the first one? Yeah, less than 98% SNPs, yeah. And so this left 9,748 SNPs for 237 samples. And we got similar groupings to our split tree. So tetrapleura, most of the samples clustered together on one side of the PCA, and on the other end of the PCA, fibrosa, and we also got sidrifloa and fusiformis on the other corner of the PCA. We got a tight group of samples in between fibrosa and tetrapleura, which we named here putative hybrids. And we got this weird smear of samples between sidrifloa and tetrapleura, which we also named as putative hybrids. And then we kind of got these samples sort of in between all three, and we called those putative hybrids as well. So we did a LEA analysis to investigate the structure of all of our data and samples, and basically this uses the SMNMF function, and this shows K equals 3, and here are all the Eucalyptus fibrosa samples, so in the green, and you can see that most of them have strong assignment to Eucalyptus fibrosa. There were a couple that were actually hybrids there, or come out as admixture. Then most of the sidrifloa samples also had strong assignment to sidrifloa, except for this one here that's clearly a misidentification and was actually fibrosa. And then fusiformis is interesting because while it does have a strong assignment to the sidrifloa group in blue, it also seems to have admixture from fibrosa and from tetrapleura. For the samples of Eucalyptus tetrapleura that we collected, these ones here were collected from the northern part of the distribution of the species, and you can see that they're clearly admixed between fibrosa and tetrapleura. Another northern population came out with admixture from sidrifloa, tetrapleura, and fibrosa. And then the rest, you can see there were a few misidentifications where we thought we sampled tetrapleura, but in actual fact were fibrosa, and a couple of samples that we thought were tetrapleura but came out as sidrifloa. But most of these were fairly pure. I say fairly pure. The most pure were actually around Wells Crossing and the central populations of Eucalyptus tetrapleura, which was where they did most of the clearing for the upgrade to the Pacific Highway. So we clearly got admixture in our layer analysis. So I wanted to investigate patterns of hybridization further. So I did a tree mix analysis. And for those of you who aren't familiar with tree mix, basically what the software does is it generates a maximum likelihood tree, and then you can add migration events to the tree, and the software will tell you which lineages are most likely to be admixing. So in this we have a maximum likelihood tree showing Hydrofloa in one clade, so all the populations there, and Fusiformis is sort of off here. Fibrosis in another clade down here, and Tetrapleura populations, most of them are here, although those admixed populations have come out in a separate clade, sort of down here. So you've got one clade there for one of the northern populations, and the other 4 northern populations are here. And basically the arrows in the trees suggest gene flow between lineages, and they're weighted by colour. So the redder the arrow, the more gene flow there is between the lineages. So here we've got gene flow, quite strong gene flow, the analysis suggests from Hydrofloa into one of the northern populations of Tetrapleura, which came out as admixed. And similarly, there's also strong gene flow from Fibrosa into the other northern population. And then we've got weaker gene flow from, I think, Fibrosa into Tetrapleura from Tyndale Road, and then we've got weaker gene flow from Hydrofloa into the central populations of Eucalyptus tetrapleura. And you also get a tree mix also produces a heat map, and in the heat map, basically the bluer or blacker colours between populations suggest likely candidates for recent admixture. So here we can see that while there are a few populations that are likely to be recent candidates for admixture, a lot of them aren't, so perhaps that suggests there's been historic admixture in the species. So in order to identify if what we were seeing were actually hybrids, we ran the new hybrids software, and that was quite complicated because we had to use subsets of the data. But so for simplicity's sake, I'm just presenting on the map what came out as pure Tetrapleura and what came out as hybrids between Tetrapleura and Fibrosa and between Tetrapleura and Hydrofloa. And basically what we found, so the red indicates pure Tetrapleura, the grey indicates hybrid between Fibrosa and Tetrapleura, and the black indicates hybrid between Hydrofloa and Tetrapleura. So basically what we found was that, consistent with our other analyses, everything to the north of the Clarence River was actually a hybrid, and these came out as F2 hybrids. And everything below the Clarence River was a mixture between Tetrapleura and hybrids. And this made a lot of sense with a lot of other studies that have been done on the north coast, where the Clarence River has been shown to be a strong biogeographic barrier. And in regards to the south of the Clarence River, what you can see is that on the western side, there's hybrids and pure individuals. On the eastern side, there are hybrids and pure individuals. And the purest populations are really around that Pacific Highway where they did the clearing. So overall, we found an unexpected restriction in the range of Eucalyptus tetrapleura, because in AVH, these are all listed as pure tetrapleura. We found that there were issues with misidentification and morphology. To illustrate, both these photographs are taken from collections up there, and we identified that as Fibrosa and that as tetrapleura, yet they've come out as the same genetically. So given that there's extensive, or at least we found evidence of extensive admixture, and that there's been a history of high levels of disturbance, we consider tetrapleura to be at risk of genetic swamping. So we recommended that the remaining populations be preserved and disturbance minimised of Eucalyptus tetrapleura. We also recommended that new populations be established in suitable local habitats, that seed be collected from as many pure individuals and populations as possible, and that given that you can source seed from a tree, but you don't actually know if the propagules are going to be pure or not, so we recommended genotyping the seedlings collected, and also to monitor those seedlings, and in the longer term, it is likely, following on what a lot of other people have said, it's likely that long-term monitoring will be needed on the natural populations and the planted stands. So, like all research projects, it's opened up more questions. For example, what is Eucalyptus fusiformis? So, given that we don't know, it'd be good if a wider study is done on Eucalyptus fusiformis. And looking to the future, restore and renew, you've already heard quite a lot about that from Jason's really good talk. So we're looking at doing landscape genomics of a lot of eucalypt species, and we're learning from the Eucalypt tetraflora experience to apply to this experiment. And thank you for listening, and I'd like to acknowledge all the people that helped me, including my collaborators on this project, Peter Wilson, Rob Koyman, Maurizio Rosetto, Marlene Vandermeer, and also Jason for writing the incredibly helpful codes that have enabled us to analyse all the data, and also John O'Donnell from Roads and Maritime Services for helping with the fieldwork, and also Rose, because I've sent her an email of some of these data analyses and she did provide lots of comments, so thank you, and yeah.
Time for one quick question. Renee?
Maybe a tough question, I'm not sure. So why do you discriminate against hybrids?
I don't discriminate hybrids at all. I actually think hybrids are really important, and everything. I was just more about how they basically cleared through the central population, and they cleared through what was the more pure Eucalypt tetraflora, and they were basically setting up one offset site where they wanted to allow it to naturally regenerate. So yeah, in our advice we were just saying, well, you probably should have more offset properties to preserve those. I will say that the hybrids that we identified on the north of the Clarence River, a lot of those are in protected areas, so they are protected for the longer term.
No, thank you. Thanks, Susan. Now, the next speaker is Rebecca Jones from University of Tasmania. Again, continuing the theme of hybridisation.
Margaret. So for this project I was tasked with combining some data sets that were generated over a number of years with honours students that we had over the years. Some of the data is getting a bit old. I will be presenting some microsat data too, so thank you, Linda, for also doing so I don't feel too guilty that there's not any genomics in this. But I really like this project because it brings together different data types. So nuclear data, chloroplast data, glasshouse grown seedlings, and some long-term common garden experiments to look at to show that the interplay of hybridisation and selection have shaped the patterns of variation that we've seen in these endemic Tasmanian eucalypts. So the alpine white gums are a group of species that are only found in Tasmania. Eucalyptus archeri, Gunnii, and Onigera occur on the central plateau where there's complex patterns of clinal variation that link them. And then Gunnii and Onigera also occur down in the southeast where when they co-occur there, they're quite distinct. And Cordata and Marisbii are found in the lowland regions. Marisbii is probably one of the rarest eucalypts with just two main populations left, and that's the one that's been buzzing away sequencing right now as one of the two species that you guys are sequencing this week upstairs. Well, it started off upstairs in our lab. So this species complex is a really good one for studying the evolutionary processes that occur when populations come back into contact after glaciations, because it occurs, three of them occur in the Central Highlands, which was thought to be treeless during the last glaciation, and they also occur down in the southeast, which was a glacial refusion. So we can compare the patterns of variation that we see in those two environments. So as I said, that's their current distribution. As I said, the Central Highlands area was thought to be uninhabitable by trees in the last glaciation. And so I'll show, and so these populations that are there now have come back into contact there. And I'll show that the complex patterns of variation, that interplay of hybridisation and selection today, and I already told you the types of data that let us come to that conclusion. So we sampled populations from across the distribution of these taxa. We particularly concentrated on the Central Plateau region, following the clines that link Eucalyptus archeri and gunnii. So archeri in the north, down into Eucalyptus gunnii subspecies divaricata, which you'll meet on the field trip later this week. And so that's from the sort of green morphology of the stems and leaves down into the glaucous morphology in subspecies divaricata down that latitudinal cline, and then a roughly east-west cline that links subspecies divaricata and subspecies gunnii that is an altitudinal cline. So we sequenced the JLA region of the chloroplast genome, and this map just shows the haplotypes that were found at each of these populations, and they're coloured. Each colour's a different haplotype, but the similar kind of colours, the bluish colours, indicate similar groups of haplotypes. And so 3 key results emerged from that. First of all, we found no overlap between the regions, comparing the putative glacial refugium with the recolonised or newly expanded area on the plateau. And that's something we've shown before, and we've talked about that already in other eucalypts this week. Secondly, though, we found really low genetic diversity, haplotype diversity in the Central Highlands, only 5 haplotypes found across that whole region, compared with 39 found in the glacial refugium. And again, that's no surprise, that's what we expect to see. We saw extensive patterns of sharing on the Central Plateau, and this occurred to a lesser extent in the southeast. But what was quite interesting is when we looked at the nuclear microsatellite diversity, there were, and as measured by allelic richness here, we found high levels of diversity on the plateau compared to the southeast, and this is independent of population size. And so this could be a signal of these, so this could be a signal of a suture zone where these populations are coming back into contact, and so previously separated populations from refugia elsewhere coming back into contact in this area. And this was accompanied by very low genetic differentiation among the populations on the plateau. So, I mean, this number here, 0.025, remember this covers 3 morphologically distinct species in this area, so there's very low differentiation. So that's further evidence that there's quite extensive pollen flow, or has been extensive pollen flow among these populations in this area. Compared with the southeast, so gunnii and Ernidra occur in the southeast, and there's a higher FST among those populations down there. And also when we compare the pairwise FST between co-occurring populations of gunnii and archeri in the Central Plateau versus the southeast, we see a big increase in FST in the southeast. This is further illustrated in this structure plot. I've put a box around the Central Plateau populations, and you can see it's a real mixing pot. So this is a bit of an indicator that the morphological variation that pulls apart these 3 species could be due to selection. I should also mention, of course, since we did this, we've been able to use dart markers and things like that. So we have thrown a subsample of some of these through with some dart markers, and they were also really poorly resolved there. So I think that it is, even though this is just with 13 microsats, I think this is a real pattern that we're seeing, that these species are really poorly resolved. I've used structure quite a lot, but I haven't ever used the Lock-Pryor method, but I read that that's a good one to use when you get this really low differentiation. So so the Lock-Pryor method includes some location information for those populations. So I ran that, and we did manage to tease apart some gene pools there, and so it does look like there's these 3 gene pools coming into contact on the plateau. We've got a gene pool that corresponds to the northern archeri and gunnii in blue. We've got a southern gunnii gene pool and an Ernidra gene pool. But on the central plateau, there is a mixing pot where these populations meet. And we do see some evidence here of potentially introgression from Eucalyptus ernidra into, so the letters here are the morphology of each population. And we potentially see some introgression from ernidra in red into Gunnii subspecies Divaricata here that could indicate that that taxon is the result of hybridisation from ernidra into Gunnii, and so potentially some genes from ernidra, which is adapted to a drier environment, is what enables subspecies Divaricata to occupy its quite distinct environment, which is not only cold like the other Gunnii but also dry. And so also here though we were unable to pull apart Archeri and Gunnii in the north, so they're still weakly differentiated even though they've got morphological differences. So this does imply there's selection on the morphology. And when we grow these populations in the glasshouse, these species are indeed distinct. So it's not just environmental variation across that cline causing the differences in morphology, there is genetic variation there as well. And so, yeah, the species are morphologically distinct. We've got Archeri, Gunnii, ernidra clustering, and then the intermediate a couple of intermediate populations that were sampled in that cline do fall in between. And Peter Harrison fitted in the climatic variables, and this variation does appear to be associated with environmental variation. I don't need you to look at this table, but the formal test that these selection is driving these patterns that we see is that we found a significantly higher QST than the max FST for many of the traits that were measured. So it is indicating that selection is driving these the speciation patterns on the plateau. And further evidence of divergent selection shaping the patterns that we see come from these reciprocal common garden trials that were set up in the Central Highlands by Brad during his PhD. So they're 40-year-old common gardens. They do have pretty small sample size, so we do need to take these results with a grain of salt. But they're still really impressive because they've been growing for such a long time. And so there are 4 gardens indicated by the stars here, and the local genetic material at each of those sites is indicated in the colour, and so all 5 of those provenances are grown at each of these 4 common environments. And we found good evidence of local adaptation in these gardens, so that there's adaptation going on in this Central Plateau region. We generally found that the local provenance outperformed the non-local provenances, as you can see here. And if we look at the cline that links Archeri and Gunnii, and so at Projection Bluff High here, where Archeri is local, the 2 Archeri provenances outperformed the Gunnii provenances. And at the other end of that cline, where Gunnii is local, the 3 Gunnii provenances outperformed the Archeri provenances. When we look at the cline that links Gunnii subspecies Gunnii with subspecies Divaricata, it seems that it's asymmetrical in this case. So at Pensford, where subspecies Gunnii is local, this subspecies outperforms the Divaricata, but I mean it has died off, it died later. But at the other end of that cline, at the top of that cline, the cooler site, the provenances are doing about as well as each other. And this might be an indication, we might be seeing the results of climate change here, because the temperature at Lyawini High is of course increasing, as it is everywhere, and it's heading towards the temperature at that the Pensford provenance would have experienced in the past, and that it must be could be adapted to, and so this could explain why these provenances are doing about the same, even though this is a low altitude provenance now growing at a higher altitude site. So in summary, we found that there's this interplay of hybridisation and selection that's formed this response to past climate change. And I do want to make the point about the importance of maintaining species interactions under future climate change. So allowing hybridisation to occur, because that hybridisation could inject diversity on which selection can act, and even as Brad described this morning, potentially allow species to migrate using hybridisation as a dispersal mechanism. So that's all. I'll leave you with a picture of subspecies divaricata. You'll see this beautifully depressing image yourselves out the bus window this week as we're going to visit subspecies divaricata on the plateau, which is the most frost-tolerant eucalypt, and sadly we're losing that genetic diversity. Luckily it's growing in France, so we won't lose it entirely.
About genetic swamping, you pitched the opposite in terms of the hybridisation and the diversity providing adaptive variants.
Yeah.
There would be a case of genetic swamping here that anyone could make?
Yeah, so I'll put the proviso then. You do need to keep an eye on it. So I like the idea of conserving hybrids, and I'll move on to thinking about Eucalyptus marisbii, which is in massive decline. And we do see hybrids with Viminalis. They hybridise quite readily. And the question is, and it was actually a question posed to us by the Department of Primary Industries here, do we chop out those hybrids to allow it to be a pure kind of site? And I think not. I think it could be injecting genes that can be useful for adaptation, but I think it's something you need to keep an eye on. And also make sure you have really good ex situ conservation strategies in place, so you have that pure genetic material if you change your mind later on, and want to use good pure genetic material.
Thank you. Thanks, Rebecca. So our next speaker is Peter Harrison, and we're going to do a quick switch back to talking about adaptation.
Excellent. Thanks very much, Margaret, and thanks everyone. So yes, I'm just going to give a very quick overview of the different ways that we can introduce genetics into climate models to delineate adaptive seed zones. So as we've heard a fair bit so far, we can incorporate adaptive genomic variation into climate models using various different methods. And one method that we've used here, which is work from Dot Steen, as well as what Margaret mentioned earlier, was Eucalyptus tricarpa, where we used a cap-based approach. Sorry. Yeah. Oh, that, yeah. Cool. Yeah. We can grab from this analysis, we grabbed the first canonical axis, which was represented arid adaptations across its distribution. And as we can see here in the red, this is the arid adapted genotypes, which are more arid than these mesic adapted. But this isn't the only way that we can introduce genetics into these climate models. We can also use traits under divergent selection. So to explore this approach, we used 20 seedling traits grown in a glasshouse experiment for 893 seedlings from 312 families sampled across 45 populations of Eucalyptus ovata in its distribution in Tasmania. So our first step was to filter down these 20 seedling traits to those which showed significant provenance differentiation, significant divergent selection, association with some facet of the environment, and genetic independence from other seedling traits. So as we filtered all our morphological traits through this, we ended up with only 8 seedling traits and one of which was relative lignotuber size, which increases linearly with maximum temperature, which is similar to what Dot showed for Eucalyptus falciflora. We then took those traits and summarised the population variation, and we did this for using a matrix of generalised distance among the provenances. We then put this matrix into a cap analysis, but that's only just one option, and associated that with 7 independent facets of the environment. We then can grab that cap index and then project it in and create these interpolated adaptive surfaces. So we mapped out this first canonical index, which represented adaptation to aridity. And as we can see, there was an epicentre of arid adapted provenances in the Midlands. And this little black cross is one of our restoration sites. And as we can see, it's right on the margin of this arid adapted zone. When we project this into the future, we can see that the selection surface, there is potentially a large proportion of the northern Midlands that will be outside the adaptive range of this species. And our restoration site may be potentially at the adaptive limits of this species. So we can actually use these surfaces in an applied sense to help identify where we might choose our provenances for seed collections. So if we were doing restoration in the southeast here, based on its predicted selection, we may be best to supplement the local seed with seed collected from these arid adapted zones. But we can also grab the climate weights from these associations and then feed them into specialised seed sourcing packages, and then be able to properly target provenances. So, thank you.
See you over there. So Sakshi's going to talk from Greening Australia and La Trobe. We're going to talk about guidelines for habitat restoration in the changing climate.
Hi, thanks. My name's Sasha. I'm from Greening Australia and I also work at La Trobe Uni. I'm going to talk today about a project which has just started up, so I have no data. I'm just going to explain the methods behind the project. It's a project which is funded by the Victorian government, and it's trying to develop climate-adjusted provenancing plot network across Victoria. So, what's plot network objectives? Well, it's to enhance habitat resilience in an uncertain and unpredictable climatic events, and try and incorporate genetic and species diversity into those restoration plantings. And we really want to inform land managers how to build resilience to climate change during habitat restoration. So part of the project is looking at using these plots as research areas, but we're also working really strongly with catchment management authorities and land care groups to hopefully use these plots as seed production areas into the future as well. So they've got 2 main goals. So what are the key steps when developing a climate plot network in Victoria? Well, we want to select on environmental gradients. We've heard today in a lot of talks that things like aridity and extreme temperature and rainfall events are really important factors to take into account. We want to prioritise the species we use in revegetation, and we also want to look at what plant provenances we use in these revegetation activities. So firstly, if we're looking at environmental gradients and where to locate the plots, this is an aridity gradient across Victoria, and if we had 12 plots across Victoria, we could hopefully sample all of these different aridity gradients. And equally, if we looked at mean maximum temperature for the hottest month, this is in 2070 projections, this would also show that these plots would also capture that difference across the whole state. So ideally we'd have 3 sites in very high aridity and temperature zones, high aridity and temperature zones, moderate and low aridity and temperature zones. And what are our criteria for selecting species? Well, we want species which are widely distributed across the state and are commonly used in revegetation projects at the moment. And we also want species which are keystone in the landscape, which are really important for that landscape. And we want a mix of different life forms in these plantings. So some species which are commonly found across Victoria, Eucalyptus viminalis, Eucalyptus commendalensis, Lamandra longifolia and bull oak in this slide. And these are roughly 10 of the most commonly used revegetation species used across Victoria. There's other species which aren't in there, such as Poa lab, but generally speaking, these species are used very often in revegetation projects. And we can use climate-adjusted provenancing, as has been suggested previously, and we can also try and choose different provenances, both from local areas, those which are an intermediate distance away, and those from a long distance away. So if we're looking at sites, ideally we'd have 4 hectare areas, each in those locations I showed before. In each of these areas there'd be 4,000 plants. These would be made up of 5 different species, so 800 individuals per species, and these would be from local provenance, wetter provinces, drier provinces at an intermediate distance, and drier provenances from a further distance away. And these species would be repeated across the whole landscape. As I said, this project is very much in the developmental stage, so any ideas or feedback would be greatly appreciated. And we want to monitor how the local species are maintaining the environment and survive and grow, and also how they change genetically over time. So we're not looking at this project as a short-term objective, it's got very long-term implications. And this is an example of how species might change in the future. So this is for Eucalyptus viminalis, and this is a projection to 2070 about how the species may change in that landscape. So these climate plots will hopefully capture how, well, it'll be able to tell if these species are actually dropping out of that landscape and which species are maintained, which provenances. So, thank you.
Sasha, any sort of time for a question?
How are you going at getting the science?
Good. We've got interest from a lot of catchment management authorities and land care groups. Also, universities such as La Trobe have multiple campuses across the state which potentially we could use. And also people like Vic Roads and Melbourne Water have various sites across the state which may also be available. So we're looking, so the sites we want are, I guess, preferably not on private land because we don't want them to change hands and make it like more difficult to sample from. So yeah, as I said, it's in the early stages and we don't actually have money to set up the plots as yet. But we're working on that, and we're working with Lynn Broadhurst and a group of other people nationally to try and get this plot network going, not only in Victoria but on a national scale.
Thank you. Please join me in thanking all of our speakers this afternoon for really